Hello, I'm Hansa and I'm with Akiko and Jet to present the AKT signaling pathway.
Alright, the pathway begins by the activation of the receptor tyrosine kinase, or RTK, which is located at the cell surface. RTKs can exist as two monomers, but as we'll see, will form a dimer. The way this happens is that the growth factor insulin binds to the receptor binding site of the RTKs. Once bound, it produces conformational changes to the RTK, making a dimer, as you can see from the figure. The RTK then autophosphorylates, and once the RTK is phosphorylated, insulin receptor substrate, or IRS, may bind to the RTK. The IRS recruits PI3 kinase, or PI3K, and PI3K has the ability to phosphorylate PIP2 to PIP3.
At the plasma membrane, where PIP2 and PIP3 are located, PI3K has to phosphorylate enough PIP2 and thereby increase the concentration of PIP3. PIP3 then recruits other proteins towards the plasma membrane, including AKT1. However, our figure doesn't show that PIP3 directly activating AKT1, because AKT1 needs other substrates to be fully activated. PIP3 is also responsible for recruiting these necessary substrates, such as PDK1, for full activation of AKT1.
Fully activated AKT1 is a downstream activator with many substrates and effects on cell processes. One of the effects is related to the tumor suppressor protein called FOXO. FOXO is a substrate of the enzyme ubiquitin ligase, which attaches ubiquitin peptides onto proteins for degradation. Through phosphorylation, AKT1 inhibits FOXO and blocks FOXO from entering the nucleus. In other words, AKT1 lowers the concentration of the protein FOXO by phosphorylation. For this reason, AKT1 is an oncogene as it prevents ubiquitination, instead promoting cell proliferation. Therefore, AKT1 contributes to sustaining proliferative signaling, one of the hallmarks of cancer. And here are our questions. So number 1 is, does phosphorylation of FOXO by AKT1 increase or decrease cell proliferation? And number 2, what molecule does PI3K phosphorylate in order to activate PDK1? And 3, does insulin directly activate receptor tyrosine kinases? What is the role of insulin in activating RTKs? 4, if ATK1 inhibits box molecules, does inhibition of ATK1 increase/decrease apoptosis? And 5, what modifications to PIP3 can result in the deactivation of the AKT signaling pathway? And these are the references that our group have looked up. Thank you for watching our presentation and have a nice day.